There's another important metabolite called uracil that we described already in the past to be oscillating very beautifully in the normal chow. You can see that oscillation is strongly dampened, and this is of course critical for DNA synthesis in DNA metabolism. Now, the surprise came from this experiment. This is now transcriptome analysis, and this is now a normal chow experiment, and this is a high-fat diet. You can see about 1,000 genes here, beautifully oscillating by groups. You can see there are different groups oscillating different peaks, different Zeitgebers. The oscillations of those is dramatically disrupted. That was that was like, let's say, expected. We were not surprised to see this, but we were surprised to see something else. When you're going to take now another group of genes, this is another 1,000 genes that will be normally not oscillating in normal chow, they start to be oscillating a high-fat diet. So what a high-fat diet is doing is not to disrupt, it's not simply to disrupt the normal cycles of our physiology, but is installing a new program. There's a new clock that is now starting to click and to tick in the high-fat diet liver, and this is now a new type of clock, and of course we know precisely what those genes are. And you can imagine this being part of the inflammasome at least. So I'm not going to go through too many details, but I decided to show you two examples. These are two genes of this group, SIDEC and PCX. And we wonder about how is it possible that genes that were not oscillating before now start to be oscillating? Do they have E-boxes? Do they have the normal sites where clock and BMAL will generally bind? The answer is no, they don't have clock and BMAL. They have other promoter sites, and what we found, and this is only part of the story, but at least in one case, the high-fat diet that disrupts circadian function in the clock is, okay, I'm going to skip this, is controlled by PPAR gamma. Anyway, so the picture is the following. Loss of oscillation, we have two pictures. A loss of oscillation, those are all the transcripts who will start to not oscillate anymore in high-fat diet. Those ones do not oscillate anymore because clock and BMAL are not able to be recruited to a promoter anymore, and thereby the oscillation-specific metabolite becomes flat. That is the example of NAD I showed you. So normally, clock and BMAL controls the nicotinamide phosphoribosyltransferase transferase, is the enzyme critical for NAD synthesis. That's a normal situation. In a high-fat diet, clock and BMAL are not recruited to that promoter, and thereby NAD becomes flat. In the case of a gain of oscillation, now that all those genes I showed you to become oscillating, at least one player is PPAR gamma, a critical element of differentiation of adipocytes, and as well as a critical element in inflammatory response. In normal chow, PPAR gamma protein is flat, and thereby some key genes controlled normally by PPAR gamma are not oscillating, and thereby also the metabolite controlled by these genes, SAHA, critical enzyme, critical metabolite from the metabolism methylation, is flat. In a high-fat diet, PPAR gamma becomes oscillating, the gene becomes oscillating, and otherwise also the metabolite becomes oscillating. So PPAR gamma is recruited in a circular fashion. All right, summary, basically what I showed you is that nutrition is critical for our physiology, as we all know. I think the next speaker is going to tell you how important it is the way our nutrition is changing our society. We've got to be very careful of what we eat and how much we do that, but also when we do that, because the way we get food, it will challenge our system. And if you have the wrong food, that might reprogram the circadian clock. It's not just a matter of disruption, but we'll reprogram it in a way that will make it different and will make it more towards an inflammatory type of situation. The reprogramming involves a number of pathways. We unraveled and coupled that will be published soon. As I said, these include both epigenetic and transcriptional pathways. Reprogramming is nutrition-driven, is independent of obesity. I didn't show this data to you, but also we found that The clock is very plastic, and in fact, our reprogramming is reversible. We can put this mice now in alpha diet back in normal chow, and the previous program will be starting it. And of course, as a last point, this tells you that depending on the nutritional epigenetic context, the circadian epigenome and the magnitude of the circadian clock is controlled by the circadian machinery might change and become more expanded. So just to finish, I hope I convey to you that the connection between nutrition and metabolism and epigenetics, and thereby how sometime we believe that this effect could be even transmitted through generations. We believe that a good chunk of that connection is played by the circadian clock machinery. I'd like to thank the people in the lab. Again, this is not far away from the campus, but mostly I'd like to thank Selma and Kristen right here, although everybody really contributed. Thank you for your attention.
Okay, thank you. Are there questions from the audience? Anyone? I guess one question that I think of is, this is probably really, high-fat diet perturbs the redox state, and so the question is, how much is oxidative stress, impacting this either as triggering some of these events or perhaps being downstream? I imagine with these major changes in metabolism that you're going to have major effects on redox signaling.
No, of course, we do know that very well. In fact, the genes that we see activated, as well as the metabolites that we see now changing, reflect that picture. What we were not expecting was the possibility of the plasticity of the system to be readjusting to a new type of cycle. That's to say, do we expect the obese people are over fed, will be disrupted in their circadian cycle? They're not disrupted. They are different. That's the concept. And I believe that's an important concept because from a pure pharmacological point of view, I will imagine different type of strategies for a different type of people.
Is it fat or sugar? Is it fat or sugar in these diets?
I cannot hear you.
I'm just wondering whether it's the sugar or the fat in the diet that actually made a difference.
That's a good question. My picture right now is sugar. The reason I'm telling you this is because we've done some experiment with ketogenic diet, which is high in fat and low in carb, and we don't have the same type of picture. So I believe different type of diet are going to induce different type of reprogramming.
I have questions. As you mentioned that there is a relation between nutrition and epigenetics. Can you comment if there is any relation between Prader-Willi syndromes and the clock center, I think?
What did you say?
Prader-Willi syndrome.
Sure. No, there is one, of course. Prader-Willi syndrome is one of those diseases that has a very strong epigenetic basis. Now, whether that has a connection with the disruption of the clock, there was a couple of reports showing that in mouse models, showing that, unfortunately, those reports are only limited in quality, so that the only thing these people did was to look at the motorical activity of those mice. But yes, they did have some disruption of the clock system.
I want to know what is your opinion about paleo diet and no sugar, no grain diet?
I could really cannot understand. What is my opinion or what?
About paleo diet or no sugar, no grain diet. Only fruits and vegetables.
So if it's good to eat fruit and vegetables, that's it. That's the question. Yeah, it is. It is good. I cannot hear the question properly. But, so if it's good to eat fruit and vegetables, that's it. That's the question. Yeah, it is. It is good. I cannot hear the question properly. But, so if it's good to eat fruit and vegetables, that's it. That's the question. Yeah, it is good. I cannot hear the question properly. But, so if it's good to eat fruit and vegetables, that's it. That's the question. Yeah, it is. It is good. I cannot hear the question properly. But, I'm not a nutritionist. I can only say that a balanced diet is the best thing you can do. And definitely, vegetables such as broccoli and so on are critical antioxidants. So definitely you can go that way.
Okay,
Sorry, one more question if I may.
last question.
Following on from the previous question, may I please ask you what your opinion is about a diet rich in fruits and vegetables and potential effects of phytochemicals? For example, compounds such as resveratrol might have on SIRT1 activity and NAD levels in the long term, comparing a high-fat, high-sugar diet, which is processed, and a traditional, healthy, and balanced diet full of fruits and vegetables.
All right, so that's an interesting question. Two things to say. One, I believe the next speaker will be more helpful in that direction. The other is that we are thinking that in that sense, with experiments in the lab, and we found that a diet that is rich in some antioxidants. Okay. In some antioxidants will be actually positive for sirtuin activity.
Okay, thanks for a great talk, and the co-chair will introduce the next speaker.
So it is my pleasure to introduce Prof. Franco. He has a major contribution to molecular epidemiology, including genetic association with diseases such as diabetes. In addition to genetics approach, he has made important contribution to our understanding of how particular class of both endogenous and metabolite can has effect on the disease susceptibility. Prof. Frank will deliver his talk about nutritional genomics and metabolic in obesity and type 2 diabetes. Welcome, Prof.
Okay, thank you. Thank you for your kind introduction. Let me start with this concept of systems epidemiology. It's basically application of systems biology to human epidemiological studies or clinical trials. In the past several years in the field of epidemiology, there is a major paradigm shift from this so-called chronic, so-called black box epidemiology to systems biology by incorporating omics technologies into large population-based epidemiological studies and clinical trials. So this approach allows us to investigate the interactions between exposome, which is all non-genetic elements to which we are exposed, and the quantifiable element of human physiology. So here, exposome include various factors we are exposed, as I just the last speaker mentioned, disrupt sleep is a very important factor that may induce metabolic changes, dietary intakes, smoking, obesity, diet, other dietary factors, exercise, and so on and so forth. So diet is one of the most important environmental factors that we have been studying. And so the area of nutrigenomics is the study of the interplays or interactions between dietary factors and human genome, epigenome, transcriptome, metabolome, and the metagenome. So our goal in nutrigenomics is to study how dietary factors modulate gene expressions, epigenetic changes, and metabolic changes. We are also interested in how genetic background can modify the effects of diet, weight loss, and other phenotypic outcomes. So I just want to give you an example of how this approach can be applied to diet and health outcomes. In the past 10 years, more than 30 large epidemiological studies have found that people who drink coffee on a regular basis have significantly lower risk of developing type 2 diabetes. So I guess this is a good news for some people here. This is a meta-analysis published a couple of years ago. As you can see that as coffee consumption increases, the risk of type 2 diabetes decreases in the dose response manner. And this has been found in numerous studies in different populations, and the results are very robust. And at this point, we still don't know what are the biological mechanisms underlying this protective effects of coffee against diabetes. Coffee is a very complex beverage. It contains hundreds, perhaps thousands of bioactive compounds. So in this situation, the nutrient genomics approach can help us better understand the biological mechanism underlying this epidemiological observation. So here I illustrate several different nutrient genomics approaches to the relationship between coffee and diabetes. So at genomics level, recent GWAS studies have identified several genetic variants that contribute to coffee intake. I will come back to this in just 1 minute. And then gene expression studies in animals and humans have found that coffee consumption can actually downregulate multiple pro-inflammatory genes, gene expressions in the various tissues, including adipose tissue, liver, and muscles. And also, small-scale targeted studies in humans have found that coffee consumption reduce plasma concentrations of pro-inflammatory cytokines such as CRP, IL-6, featuring A. And recent metabolomic studies have identified several metabolites, caffeine metabolites, and also other components of coffee metabolites that predict decreased risk of type 2 diabetes. So those metabolites can be measured in either plasma or urine. So those studies at different levels clearly contribute to our understanding of the biological mechanisms underlying the relationship between coffee consumption and reduced risk of diabetes observed in numerous epidemiological studies. This is a GWAS study conducted by a postdoc in our group, Maryland colonists. It was published a couple years ago. This study included more than 47,000 men and women 50 to 60 years old. As you can see that two genetic variants reach genome-wide significance, and both loci are actually related to caffeine metabolism. And several other studies have replicated those results, and this finding suggests that genetic variation in caffeine metabolism is associated with coffee or caffeine intake. So some people have fast metabolism genotypes, other people have slow metabolism genotypes, and the different genotypes clearly determine the amount of coffee or caffeine people consume. Those studies also demonstrate that it is feasible to use GWAS to identify genetic determinants of behavior traits, such as various dietary behaviors. And we know that those behaviors are not easy to measure, and typically they are measured with errors. And the genetic variants identified from this type of studies can be used for gene-environment interaction studies. Some studies have shown that if you carry a slow metabolism, caffeine metabolism genotype, you are more susceptible to detrimental effects of high caffeine intake on cardiovascular disease and other disease outcomes. If you carry fast caffeine metabolism genotypes, you are less susceptible to the potential deleterious effects of high caffeine intake. So in this situation, genotype modulate the effects of a dietary factor on disease outcome. And also, those genetic variants can be used as instrumental variables in Mendelian randomization analysis to establish the causal relationship between this dietary factor and the health outcomes. As has already been alluded to, recent advances in metabolomics technology have led to our better understanding of metabolic pathways through which various factors, including diet, influence various phenotypes. And metabolomics is particularly relevant for nutrient genomics studies because it can help us reveal the interconnections of multiple metabolic pathways, can generate a metabolic profile of organs, tissues, and can monitor gene-environment interactions, identify subphenotypes linked with hormone regulation pathways, and also can be useful in identifying functions of non-genes. So to a large degree, metabolome is the end product of gene-environment interactions because it can be directly influenced by what we eat, what kind of medications we take, the exercise we do, pollution, bacteria, and our behaviors. This is a really groundbreaking study published by a colleague at Mass General Hospital and Harvard Medical School, Tommy Wang. This was published in Nature Medicine a couple of years ago. They identified several metabolomic signatures as predictors of future risk of developing type 2 diabetes. So this study was conducted using a very large cohort study called the Framingham offspring study. And they found that plasma concentrations of branched-chain amino acids, BCAAs, strongly predict future risk of developing type 2 diabetes. So you can see that those in the highest quartile of BCAAs have more than threefold increased risk of developing type 2 diabetes compared to those in the lowest quartile. In the same cohort, they found that BCAAs is significantly associated with increased waist circumference, increased insulin resistance, increased triglycerides, and decreased HDL levels. So those are all important components of the metabolic syndrome. This is a very interesting study published by Stan Hazen from Cleveland Clinic. He identified gut flora metabolites that promote atherosclerosis and increased risk of cardiovascular disease in animal models and also in humans. So this gut flora metabolite is called TMAO, and TMAO is derived from dietary components of choline and L-carnitine. So choline and L-carnitine metabolized by gut flora and then is converted to TMAO. And in animal models, higher level of TMAO was shown to promote atherosclerosis. And in human studies, higher plasma concentrations of TMAO is associated with increased risk of cardiovascular outcomes. And those two dietary components, choline and L-carnitine, are actually derived from animal products, especially red meat. So this study, I think, provides very neat biological mechanism underlying the relationship between high red meat consumption and increased risk of cardiovascular disease, which has been observed in many epidemiological studies. In the past 15, 20 years, we have conducted very detailed analysis on interactions between dietary factors and various genetic factors in relation to obesity, type 2 diabetes, and other outcomes. We have three very large cohort studies, this is health study, health professional follow-up study, this is health study 2. So in total, we have more than 300,000 people who have been followed for 20 to 30 years, have collected very detailed information on diet and lifestyle factors every 2 to 4 years. So far, I think we have accumulated the largest nutrition database in a very large number of participants. Recently, a colleague at Dana-Farber Hospital, the Cancer Institute, Brian Wolpert, did a metabolomic study using the serum samples stored in the nurses' health study cohort and in the health professional health study cohort. So those blood samples were collected in the early 1990s. So the participants were healthy at the time when the blood samples were collected. So the metabolomics platform that was used in this study was developed by Clary Clich at the Broad Institute of Harvard and MIT. This is a targeted platform which includes about 300 metabolites. Most of the metabolites are lipid metabolites and amino acid metabolites. So this is the Manhattan plots for the p-values of individual metabolites, showing the relationship between those metabolites and the risk of type 2 diabetes. So this is the nominal p-value threshold, 0.05, and this is the p-value threshold after adjusting for multiple testing. So many of the metabolites crossed the nominal significance threshold, and several metabolites reached significance even after adjusting for multiple testing. And this shows the magnitude of the association between individual metabolites and the risk of type 2 diabetes in our cohorts. As you can see that the strongest associations were found for branch-chain amino acids, leucine, isoleucine, and valine. So those results are very consistent with what Tommy Wang observed in the Framingham offspring study. Since we also have collected data on dietary intakes of BCAAs, we were able to look at the correlations between plasma BCAAs and the dietary BCAAs. You can see that in the age-adjusted analysis, there is a strong correlation between plasma metabolites and dietary intakes. And after adjusting for BMI, the association is still significant. So clearly, plasma BCAAs are partly determined by BCAA intakes in our diet. Then we look at the relationship between BCAA intakes and the risk of type 2 diabetes in our cohort. Two cohorts, the results are very consistent in both men and women. In the pooled analysis, higher consumption of BCAAs is strongly associated with increased risk of type 2 diabetes. As you can see that the risk was increased by more than 70%, comparing those in the highest quintile with those in the lowest quintile. So I think with the metabolomics data and the dietary data and also diabetes phenotype, we're now able to connect the dots in terms of the relationship between dietary BCAA intake and plasma concentrations of BCAA metabolites and the risk of type 2 diabetes. Let's switch gears to talk about the interaction between diet and genetic factors in relation to obesity. In the past 5 to 6 years, more than 50 genetic loci have been identified for common forms of obesity, either high BMI or high waist to hip ratio. The strongest obesity gene is FTO, and for someone who carry one risk allele of FTO, his or her risk of becoming obese is increased by 40%. However, the effects of all the other genetic variants are very modest. So you can see that the effect size is pretty small, except with the exception of FTO. And also, with all the 50 BMI or with the circumference loci combined, they only explain about 10% of heritability for obesity. So there has been a lot of questions regarding what are the reasons for the so-called missing heritability. And one potential reason that has been suggested is the gene-environment interactions, because those GWAS studies, none of those GWAS studies have considered gene-environment interactions. Last year, we published a study looking at the interactions between sugar intake and risk and genetic factors in relation to obesity in our cohorts. And we specifically focus on sugar-sweetened beverages because those are the largest sources of added sugar in the US diet, and they contribute to almost 10% calories in both children and adults. And many studies have shown that increasing consumption of sugar-sweetened beverages is an important risk factor for obesity and type 2 diabetes. Most of the sugar-sweetened beverages are soft drinks, but fruit drinks, energy drinks are also considered sugar-sweetened beverages because they contain large amount of added sugar. In the same issue of New England Journal of Medicine, it published two clinical trials of reducing sugar-sweetened beverages and prevention of childhood obesity. So both studies showed that reducing sugar-sweetened beverages can actually significantly reduce childhood obesity. So it's really remarkable that New England Journal of Medicine published all the three studies back to back on the same topic in the same issue. So in our gene sugar interaction analysis, we have two discovery cohorts in the health study, health professional follow-up study, and one replication cohort. So all the three cohorts have GWAS data. And this figure shows the relative risk of developing obesity in the next 4 years per increment of 10 risk alleles according to categories of sugar-sweetened beverage consumption. So to look at the cumulative genetic effects, we calculated genetic risk score based on 32 obesity loci identified at GWAS. So the higher the number of risk alleles, the higher the risk of becoming obese. So on average, each 10 risk alleles of the genetic risk score is associated with 40% increased risk of becoming obese in the next 4 years. However, for those who consume sugary beverages on a regular basis, their risk of becoming obese is increased by 3-fold. So, as you can see, the results are highly consistent across 3 cohorts, and also the interaction is highly statistically significant. So this result suggests that there is a strong interaction, synergistic interaction between added sugar intake and the genetic risk score in relation to future risk of becoming obese. So it means that if you drink sugary beverages on a regular basis, your risk of becoming obese magnified given the same number of risk alleles you are carrying. In other words, sugar-sweetened beverages actually exacerbated genetic risk of obesity. So I think this is really a very interesting example of how the genes and our diet can interact with each other, and an unhealthy diet can actually exacerbate the genetic risk of obesity. We still don't understand, we still don't know the underlying biological mechanisms why added high sugar intake exacerbate the genetic risk of obesity. It could be due to epigenetic mechanisms, but at this point we don't know the mechanisms underlying this interaction. In the next couple of minutes, I just want to briefly mention another area of nutrient genomics studies. So there is increasing evidence that people respond to weight loss diet very differently. So on the same low-fat or low-carbohydrate diet, some people lose a lot of weight, other people don't lose weight at all, and some people even gain weight. So one question is why there is such big individual differences in response to the same weight loss diet. So one factor could be effect modification by genetic factors. So to test the interactions between genes and the dietary interventions in relation to response to weight loss diet, my colleague Lu Qi has done a series of studies to test the interactions between certain genetic factors and the weight loss diet. In one of the studies, we used a very large weight loss intervention trial, which is pound loss. This is a very large trial, including more than 800 overweight and obese individuals. This is a 2-year randomized clinical trial. And the main study was published in New England Journal of Medicine in 2009. In this trial, the participants were randomized to 4 different diets. So you have a low-fat, high-carbohydrate diet, this is a low-fat, high-protein diet, and high-fat, low-protein diet, and this is high-fat, low-carbohydrate diet. So this shows the amount of weight loss according to 4 different diets. As you can see that the 4 hypocaloric diets results in similar weight loss. There is no significant difference across different diets. And the amount of weight loss was greatest at 6 months, but many people regained some weight over time. But as you can see, for each diet, there is pretty large inter-individual differences. So the question is why some people lose more weight, other people lose less weight, given the same diet. So in this study, we specifically look at the interaction between RS1 genotype and the diet intervention interaction. The reason we are interested in RS1 because this is a very important gene for insulin signaling and insulin sensitivity. So what we found is that for weight loss, individuals carry this CC genotype are more responsive to low-fat, high-carbohydrate diet than those who carry the CT and the TT genotype. So it means that the genotype modify the response, individual's response to low-fat diet. And for changes in HOMA or improvement in insulin sensitivity, again, the CC genotype is more responsive to the low-fat diet. For the low-carbohydrate diet, it's interesting that CT and the TT genotype is more responsive. And then we tried to replicate this result in another large weight loss study. And this study was also a 2-year randomized weight loss trial, which included more than 300 obese individuals. They were randomized to low-fat diet, low-carbohydrate Mediterranean diet. So at 6 months, the low-carbohydrate diet was most effective in terms of reducing weight. But at the end of the trial, both Mediterranean diet and low-carbohydrate diet groups lost more weight than the low-fat group. So we replicated the interaction between IS1 genotype and the dietary intervention on insulin sensitivity. So there is some suggestion that the CC genotype is more responsive to the low-fat diet, whereas the CT and the TT genotype is more responsive to the low-carbohydrate diet. And this just shows the results from the pound loss trials. So overall, the interaction patterns are very consistent between those two studies. So to summarize the results from this study, participants with CC genotype had a greater reduction in body weight and improvement in HOMA-IR than those without this genotype in the low-fat, high-carbohydrate group, and this interaction was replicated in another trial. So it means that individuals with this specific genotype may benefit more in terms of weight loss and improvement in insulin sensitivity by choosing a low-fat, high-carbohydrate diet. So this could be useful in terms of personalized nutrition. And very quickly, the second example I'm going to say is the interaction between CET genotype and the interaction with dietary interventions on blood lipids. CETP is very important for lipid metabolism because CETP inhibition has been associated with increased HDL and decreased triglycerides. And GWAS studies have identified multiple genetic variants on CETP and the increased HDL levels and the decreased triglycerides levels. So in this analysis, we look at the changes in HDL cholesterol according to CETP genotype. So for those who carry CC genotype, the high-fat diet induces much greater increase in HDL levels compared to the low-fat diet, whereas in the CAA genotype, there is no difference between those two diets in terms of HDL cholesterol, changes in HDL cholesterol. And the results are almost identical for the directed trial. In the pooled analysis, again, the genotype modified the effects of high-fat diet on HDL cholesterol increase. So people who carry the CC genotype are more responsive to the high-fat diet in terms of increasing cholesterol levels. And the pattern, the changes in triglycerides are also similar in terms of the interaction between the genotype and the dietary interventions. Again, the reduction in triglycerides is much greater, more pronounced in the CC genotype compared to the CAA genotype. And the results also replicated in the directed trial. So in summary, so in both dietary intervention studies, we found that the CETP genotype modified the effects of high-fat diet on HDL cholesterol and the triglycerides. So those individuals with CETP CC genotype might obtain greater benefit in terms of HDL cholesterol and triglycerides by choosing a low-carbohydrate, high-fat weight loss diet instead of low-fat diet. To summarize, I think the nutrient genomic studies, especially with the combination of the GWAS studies and the metabolomic studies, are very powerful tool to identify common genetic variants associated with dietary-related behavior traits, and also identify the reasons, potential genetic variants that may be responsible for the inter-individual responses to various dietary interventions. Dietary factors can alter the expressions of genes and the levels of proteins and the metabolites related to inflammation, lipid metabolism, and gut microbiome metabolism. This is the example I give to you in terms of caffeine metabolism.